Hi, in this video I am going to talk about the process of bioprospecting. The bioprospecting, as I said before, it is actually finding industrially useful, commercially valuable products from nature. It's basically an R&D process, research and development process. And the most of the bioprospecting is preclinical in nature, especially for the pharmaceutical industry. It's preclinical research to find novel compounds for the treatment. Then the preclinical stages, it is actually a very laborious process, very time consuming, because we need to screen thousands of samples to get a valuable product. If you want to get a single antibiotic, you may need to screen for 1 million microorganisms, 10 lakh microorganic colonies to get a single antibody producing microorganism. And that too is not guaranteed. Even if you screen for 1 million microorganism, you are not guaranteed to get the antibody producing bacteria. After screening 1 million microorganism, you may end up in nothing. That's also possible. So the other thing is that if you are screening a single microorganism or a single plant species, each of these samples have thousands of chemicals in it. Some of the common chemicals and some of the novel chemicals, some of the unique chemicals will be there. And if you want to study everything about these chemicals, it will become a huge study. That's why it's called a laborious study and time consuming, and it need a huge investment. And even if we study all of these chemicals, it is not guaranteed that we will find some application for these chemicals. And for the bioprospecting process, we need to follow GLP and GMP. GLP is good laboratory practices for the systematic discovery of a chemical, and GMP is good manufacturing practices. Once we found the chemical, in order to make it in large scale, we need to follow the good manufacturing practices. And expense for this bioprospecting of a single chemical is come around 800 million to 2 billion US dollars. So a industry need to invest that much amount of money to get one drug in market. 800 million to 2 billion US dollars. That's actually a huge amount of money. So the steps in bioprospecting. Bioprospecting has 10 different steps. I will go through this one. First one, field work to collect natural specimens. First thing is to get the specimens, which is also very difficult, because where to take the cell? If you are going to looking for antibiotic producing microorganism, where you are going to look? There are many places. They all around the globe we have microorganism. It's everywhere. And which one is going to have the antibiotic producing capacity? There is no guarantee this organism will produce or the other organism not producing. So all sites are potential sites. The Alexander Fleming actually found the antibiotic out of contamination, not from anywhere. It's just present there. So the natural samples can be anything. It can be a microorganism, it can be a fungus, or it can be archaebacteria, it can be a plant, animal, insect, bird, anything. So the specimen collection is also very important. In the most of case times, what people use is people look for the traditional knowledge or indigenous knowledge to assist this specimen collection. We cannot actually study all plants in the globe. We can only study a limited number of plants for these activities, the industrially useful activities. We cannot screen for all the microorganisms because the microbial diversity is so huge. So we'll limit our investigation to a particular limited number of specimens and samples. So in order to that, we should have some idea about the use of this plants and microorganisms and even animals. And once we get the natural samples, we extract the chemicals from it. We use various techniques, maybe extractions and specific extractions or chromatography, things like that can be used for the extraction of the chemicals from the specimen. Once we have the chemicals, we will look for the activities, whatever. If you can look for antibacterial activity or antifungal activity or the pigment producing activity or immunosuppressive activity. So whatever the activity you want, you can look into the chemicals. So that's what's called screening. We are screening all the samples for useful products. Okay, once we found some some samples with useful characteristics, the next step is to look for the toxicity studies. Because if you even if the chemical has the biotic potential, but if it is toxic, we cannot use it in pharmaceutical industry. So once we found the desired activity, next step is to study the toxicity of the compound. And if we found that the chemical is have no toxicity, we can actually go to the next step. If you find the chemical is toxic, you have to go back to the screening again. So if it is not toxic, what we do, we chemically analyze the chemical, the sample, to get an idea about the chemical structure and the formula. This is done to make sure that we are not rediscovering anything. Because most of the time, even if we are getting good antibacterial activity, and then we found that it is not toxic, and when we are chemically analyzing the product, it may be an antibiotic which is already in the pharmacy, it's already discovered. So in order to avoid that, the next step is chemical analysis to find the identity of the chemical. And once we found that this chemical is novel and nobody else has discovered this, we can actually go for the pharmacodynamics and pharmacokinetic studies. Pharmacodynamics is actually what the drug is doing in our body, and pharmacokinetics is what we are doing to the drug. Like, pharmacodynamics is studying how the pharmaceutical is acting in our body, how it is treating diseases, how it is killing the microorganisms, things like that. Pharmacokinetics is more like how the drug is getting absorbed, how it is distributed, how it is excreted, how it is degraded, things like that. Okay, then after the pharmacodynamics and pharmacokinetics, we'll go to the mechanism of action. We should know how the drug is acting, so we will study that. Then we can actually alter the molecular structure of the compound to get better activity. Maybe we can add some side groups, or maybe we can remove some of the chemicals atoms from the molecule to make it more active, make it less toxic, or things like that. Then once we have done it, we can go for the clinical trials. Clinical trials are actually testing the chemicals in humans. So after clinical trials, going for the regulatory approval to release the drug into the market. So these steps are actually followed in biopharmaceutical industry. I am taking it as an example. Here is the schematic diagram of what I have said before. We have natural resources. Natural resources can be anything, and you can actually either you can go for a blind screening. That means you are screening everything you come across, you can actually screen. Or you can use the knowledge of local peoples or tribals or indigenous knowledge to actually streamline or like shortlisting the screening. So you can use different samples. Once you get the samples, what we do, we will go for primary research, which is assays and screening. It is the first step. If you are looking for an antibiotic, you will run a crowd drug plate assay, and if you are looking for a pigment, you will run different types of assay. If you are looking for a gene, you will do some molecular biology techniques. So things that like that is happening in the primary screening. The primary screening is a very tedious process, because if you are searching, you need to search for many colonies or many samples to get a useful product. So once you get a useful product in the primary screening, it will go for the secondary screening. Secondary research is actually involved in toxicity test. As I said before, if the compound is toxic, you can have to actually use in the pharmacy. Okay, so we should assure that it is not toxic. Then once we found it is non-toxic, we'll go for the chemical identification. Chemical identification is to make sure that we have found a novel compound. So if you find it is a novel compound, we'll go up, otherwise we will go back. Then we'll go for more pharmacodynamics and pharmacokinetic studies. We'll do animal studies and in vitro studies for the optimization of the chemical. Once we optimize the chemical and we are sure that it is a novel chemical and it is not toxic and it has some industrial application, if we are sure about both four of the criteria, we'll go for a human trial, which is called clinical trials. It has 3 stages: stage 1, stage 2, and stage 3. And if the chemical is successful, we'll go for the approval from the regulatory bodies such as FDA. FDA is the Food and Drug Administration in US. In India, we have the similar organization, and if we get the approval, we'll go to the market. So this is the whole process. You can see that most of these processes are reversible. If you fail in the clinical trial, we'll go back to the other step. If you fail in the chemical ID method, you find that the chemical is not new, we'll actually go back to the first step. So it's a very complicated process. So in following session, we are actually going to talk about the first stage of finding the new chemicals, because it is where the bioprospecting is mostly applied. So how to find something in the nature which is useful and which can be exploited for commercial use. So we will be focusing on the primary screening of various compounds. The screening itself has two different stages: primary screening and secondary screening. Primary screening, what we are doing, we are actually looking whether the chemical is having, whether the biological resource is having any industrial application. Just mean any industrial application. Okay. In the secondary screening only, we are actually evaluating real industrial value. Okay. Primary screening is a broad screening. If you are looking for antibiotic producers, we are only looking at whether the organism is producing antibiotic or not. Okay. Nothing else will be considered. The amount of antibiotic produced, the nature of the antibiotic produced, nothing will be considered. Everything else will be considered in the secondary screening. So screening has primary screening and secondary screening. This is all process of screening. Just like the images I have shown before, we will select the natural resource and will make it either by procurement or by the preparation. Then go for the primary screening, then to the secondary screening with the characterization and structural elucidation. Then preclinical development modifications can be done. Then to the tertiary screening. It is a much simpler image, that's why I put it here. So primary screening of antibiotic. I am taking antibiotic as an example because antibiotic has a huge industrial and pharmaceutical application. And we are sure that antibiotic producing organisms are still to be discovered. So many microorganisms will be producing antibiotics in this in this world, but we are not yet discovered it. It will be just around us, maybe in your table or in your computer, or maybe in your mobile phone, or maybe in the Amazon forest or some other forest, maybe in oceans. Somewhere it will be still waiting to be discovered. So many companies are investing huge amount of money for discovering these novel antibiotics, because this novel antibiotic may be able to treat a new disease, or maybe treating a drug resistant bacteria, or maybe treating the coronavirus. Things like that can happen. So it's a huge potential. If you can come up with a medicine which is going to treat coronavirus or COVID-19, it will be a huge success in industry. So people are always looking forward to finding something like that. So I'm going to talk about a method which is called crowded plate technique, which is used for the screening of antibiotic producing microorganisms. So the steps of crowded plate technique is very simple. We can take samples from soil, water, air, wherever you want. You just take the sample and go for serial dilution. The serial dilute in such a way that each of the plates having 300 to 400 colony. Actually, when we are doing the plating, we used to say that the number of colonies per plate should be between 30 to 300. But in this case, it's crowded plate because the microorganisms should be crowded. So the dilution should be between 300 to 400 colonies per plate. Once we diluted the sample, maybe if you are taking soil, you already know, we will weigh the soil and we will dissolve in water and then go for serial dilution. Okay. Then after dilution, we'll spread it on an agar plate, then incubate it, and after incubation, we'll go for zone of incubation. Okay. As shown here, when in this plate, as we put more number of microorganisms like 300 to 400 microorganisms, each and every colonies will be very adjacent. So they will be sitting very close to each other. So if any of this microorganism is producing an antibiotic, it will inhibit all neighboring other neighboring microorganisms, which leads to the formation of a clear zone. So clear zone in a crowded plate indicating that some microorganism is producing a antibiotic. If you found some zones like that, you can actually isolate the microorganism and you can go for the further studies. So primary screening of antibiotics is done by crowded plate technique. Actually, we can do it in our lab. You take some samples and just plate it and look for the zones. If you see something, you got an antibiotic, but you are not sure that whether the antibiotic is toxic or the antibiotics is already known, but sometimes it will be a new antibiotic and chances are there always. And secondary screening of antibiotics include another technique called the giant colony technique. In giant colony technique, what we are doing, we actually plating the organism isolated from the crowded plate is plated just in this particular line, and different types of microorganism will be inoculated using a wire loop from here to here, just like like draw a line. In this, as it is a secondary screening, we are evaluating the industrial potential of this antibiotic producer microorganism. So our antibiotic producer is in the middle, and these lines indicating different microorganism. In this plate, they are actually testing many different microorganisms at a time. So one will be Escherichia coli, two maybe Klebsiella, this one may be Pseudomonas. So different organisms will be tested against our antibiotic producer. So in the secondary screening, we will know the spectrum of effect of this antibiotic, whether it is inhibiting only gram negative or it is inhibiting only gram positive, things like that. In the primary screening crowded plate technique, all you know is that the microorganism is producing an antibiotic. But in secondary screening, you know more. You know that the antibiotic is resistant. What is the spectrum of action, whether it is specific for gram positive or gram negative or for all microorganisms. So secondary screening is actually giving more information. So we will do the primary screening for all samples, but only those microorganisms show some activity in the primary screening will be selected for secondary screening. Secondary screening also include the calculation of MIC. MIC is the minimum inhibitory concentration, which is actually measure how much concentration of antibiotic is enough to inhibit the growth of microorganism. So it's done by serial dilution, just like you are doing in our lab. So you take the sample, take the actually antibiotic producing microorganism, or either you can take the antibiotic producing microorganism, or more precisely you can actually take the antibiotic, you grow the microorganism and extract the antibiotic. And what is the figure shows is that we have different tubes. All of this tube has equal number of sensitive microorganism. If the antibiotics meant against Escherichia coli or gram negative bacteria, you have to take antibiotics Escherichia coli in all these tubes in equal concentration. Okay, all the tube has equal number of Escherichia coli, and the antibiotic is serially diluted and added to this plate. So maximum concentration of antibiotic will be here, and the lowest possible concentration of antibiotic will be here. So when the concentration of antibiotic is low, its activity will be less, so the microorganism may not be getting inhibited, but at a higher concentration it will inhibit the growth of microorganism. All antibiotics are like that. It is concentration dependent. If we are using high concentration, it will inhibit the bacteria, other organism, but if in low concentration, it may not be that effective. So here is the lowest, this tube has lowest concentration of antibiotic, and this one has the maximum concentration of antibiotics. So if you are, and after incubation, if you if the tubes are incubated with the bacteria or the microorganism, you will see turbidity after incubation, inoculation and incubation. And the tube which doesn't show that much turbidity will be treated as MIC, minimum inhibitory concentration. As the antibiotic in this concentration is inhibiting the growth of microorganism, you cannot see a turbidity. But what's the thing is that if you take a sample from this this tube and plate it on an agar plate, you may see different colonies. Okay, because this at MIC the microorganisms get inhibited; they are not killed. Their growth is getting inhibited. That's enough for preventing so many diseases or treating so many diseases, but the microorganism is still alive. So another measure is that if you keep on taking samples from here, here and here, like as the concentration is increased, at some point antibiotic will actually kill all the microorganism. That concentration is called the minimum bactericidal concentration, or MBC. So in secondary screening we can actually measure MIC and MBC. Thank you so much for listening.